We're going to talk about the molecular approaches in the study of evolution. So molecular techniques are transforming evolutionary biology, so I want to talk to you guys a little bit about that. In terms of sequencing genomes, you guys have heard about that, right? The human genome has been sequenced, and the complete nucleotide sequence of a lot of different organisms has been determined. You can use comparative genomics, right, to study links between genic and organismal evolution. And generally, so far, the genomes of organisms that are sort of are at branch points or are at some place of evolutionary interest are the ones that have begun being sequenced, because in the past, sequencing was rather an expensive undertaking, but now every step in DNA sequencing is automated, mainly through the use of robots. It's getting faster all the time through technological advances. Today, at least one vertebrate genome is sequenced each year, and a new microbial genome of about 2 million base pairs is sequenced each week. And again, like I said, it allows for comparative genomics, right? Comparing living things and even using potential ancient DNA. The sea lamprey genome has been sequenced, as they are only one of two groups of extant jawless fishes. And because this organism is a pest in some areas like the Great Lakes, the genome is being studied to find ways to use genetic technology to control this organism. For example, as larvae, they're actually harmless. They're filter feeders in the environment, and it's not until they go through metamorphosis is when they get their teeth and their tongue that can bore a hole through the side of a fish. So if you can get them from, say, stopping metamorphosis, right, to inhibiting that, then you could do a good job in controlling the hole. Here's another picture. Differential gene expression. We talked a little bit about this already, but it's defined as a difference in timing, level, and location of gene expression. So you can have different genomes between organisms, right? But you can also have different gene expression between organisms. This is thought to contribute a lot more to morphological differences between species than the genome itself, right? Than do simply just substitutions, even mutations in those genomes in protein coding regions. Microarray technology right now allows us to correlate morphological differences between species with gene expression status across their entire genomes. Genes that show marked differences in expression between humans and chimpanzees are key controlling regions in the genome for brain cell activity, for example. And one thing that we've noted is that some people have called us the neotenous clan of apes, right? Neoteny is sort of the retention of childlike characteristics in mature members of a species. So humans much more closely resemble chimp or gorilla infants than adult survivor species. So you can see that there's maybe we retain those neotenous characteristics throughout our life, but chimps have a differential gene expression where they have more adult feature characteristics later in life. There's something called the neutral theory. This was proposed by Motoo Kimura. States the majority of evolutionary change in genes and chromosomes occurs via mutation by random drift. So the neutral theory suggests that most change in DNA sequences and proteins will take place in regions that would not affect the organism fitness drastically or be modified by mutations. So it's saying that a minority of evolutionary change is through adaptive mutations being driven to fixation by selection. The important thing to remember here is that this is not supported by many scientists, okay, but it's something that you may actually run into in the future, so I wanted to be sure that you knew what that is. Purifying selection occurs when mutations are selected against, right, so deleterious genes are actually eliminated. So you imagine that genes that code for very important processes need to be conserved. Okay, so genes are, for histones, for example, are among the most highly conserved genes in genomes. Virtually no substitutions, even in comparison with very distant animals. The histones are proteins around which DNA can line inflammatory and variant gene regulation. So you can see why a mutation in one of these is going to be selected against, because it probably causes potentially the non-replication or non-viability of an organism. If a deleterious gene of some kind makes it into this genes that code for histones, so they're very, very conserved. Positive selection occurs when new mutations are selected for. So fitness is increased, right? It's an advantageous mutation, so therefore it's selected for. Positive Darwinian selection is thought to have occurred if there's the number of advantageous substitutions in the gene exceeds the number of neutral substitutions. You can get rapid positive selection in genes that are involved in immune evasion by parasites. Remember we talked about the red queen hypothesis, right? With proteins, who's constantly changing the locks, and the parasite's trying to find the right key, right? So rapid evolution from there. It also occur in disease resistance, and even reproduction in abalones, which is a marine gastropod. You get rapid evolution in reproductive proteins that mediate sperm-egg recognition. So a specific protein expressed by the egg envelope of abalones is thought to play a role in reproductively isolating species that actually live together geographically. So adaptive molecular evolution primates. So at least two genes that are very important, right? That make humans a little bit what we are in terms of different from chimpanzees, right? There's at least two genes. One being the FOXP2 gene, which we've talked about a little bit before. This is important for language. Individuals with a defective copy of the FOXP2 gene are unable to select and produce the fine movements with the tongue and lips that are necessary to speak clearly. So they can be totally unintelligible to native listeners and to other family members without the disorder. They also tend to have dyslexic tendencies, difficulty processing sentences, and poor spelling and grammar. And scientists who examined the sequence evolution of the normal FOXP2 gene found only one amino acid substitution in the 130 million years separating mice and the ancestors of humans and chimps. Okay? But there's been two substitutions that occurred in the human lineage during the 6 million years since we've been separated from chimpanzees. Right? This gene is virtually invariant, meaning it does not vary, in another 28 orders of mammals that last shared a common ancestor about 100 million years ago. So those two substitutions in the last 6 million years in the FOXP2 gene has what scientists believe has made it important for our abilities to be able to communicate differ drastically from how chimpanzees can communicate through sound and language. Another gene is called MYH. We talked a little bit about this. The loss of this gene is thought to be responsible for the reduced size of chewing muscles in humans. MYH has been inactivated by a frameshift mutation that occurred about 2.4 million years ago. This mutation created the weaker jaw muscles, and these weaker jaw muscles did not require as large of attachment points on the cranium. So thus the sagittal crest was lost, which is that ridge at the top of most non-human primate skulls, and the human cranium was able to get rounder, which allowed for a larger brain. So I'm going to talk a little bit about genome structure diversity, because viral and bacterial genomes are very different from eukaryotic genomes. So viral and bacterial genomes maximize the speed of genome replication and minimize unnecessary genes. So as you can imagine, small genomes are advantageous for viruses and bacteria because they can be copied faster, allowing for rapid population growth. So this is a key adaptation in situations where there's interspecific or host-parasite competition for resources, being very intense. Some viruses and bacteria even exploit the genomes of their hosts, so which means they can make do with small genomes of their own. Okay? But in eukaryotic genomes, they're large, they harbor huge regions of non-coding, right, neutral genes, and even repeated DNA sequences. And what scientists have found is that within major groups, there's little relationship between the genome size and some organismal complexity. For example, humans, 3 billion base pairs make up human genome, while salamanders have about 50 billion base pairs. Okay, and genomes contain a great deal of non-informative, highly repetitive DNA that varies greatly in amount among species. Gene families, many genes are members of larger groups of genes called gene families, and they can be related to one another. So homologous sequences are orthologous if they were separated by a speciation event. Right? The genes belong to different species, they're part of the same gene family, and they're separated by that speciation event. Homologous sequences are paralogous if they were separated by a duplication event. So the genes often belong to the same species, right? But they're part of the same gene family, and you can get duplications that can cause them to be paralogous. Bacteria developing resistance to antibiotics. I want to talk a little bit about, right, how that occurs. First of all, antibiotics stop or interfere with a number of processes of bacteria. They can stop or cripple production of the bacterial cell wall, right, which obviously protects the bacterial cell from the external environment. They can interfere with protein synthesis, and they can cause problems with metabolic processes, right, such as the synthesis of things that bacteria need to thrive, such as folic acid and vitamin D. And they can block synthesis of DNA and RNA of the bacteria, preventing them from replicating properly. Well, bacteria come up with many ways to counter the action of antibiotics, thus antibiotic resistance. They can prevent the antibiotic from getting to its target. They do this so they can change the permeability of the membranes, for example. They can change the target. So they can sort of make themselves almost invisible, right, and more like a host cell. So the host sees the bacteria potentially as itself rather than an invader, right? So the antibiotics no longer recognize it as a bacteria. Some kinds of bacteria actually produce enzymes that destroy antibiotics completely, so that definitely helps. So how do bacteria acquire resistance to antibiotics? They can do so during transformation, right? Bacteria can join together and transfer DNA to each other. Through a transposon, you can get small pieces of DNA that can hop from DNA molecule to DNA molecule. They can scavenge DNA remnants from degraded dead bacteria, so they're actually getting to that they can actually pull out different DNA remnants, right, that may be helpful to themselves. Or they can get an extra chromosomal piece of DNA called a plasmid and code resistance stimulated. So thus, plasmid is kind of used here in this cartoon, right? And where it says, hey kid, want to be a superbug? Get some of this in your genome. Even penicillin won't be able to harm you. So it's easy for antibiotic or bacteria to get resistance to antibiotics. So we're going to talk a little bit about evolution of a virus. So viruses are really composed of genetic material, protein, and lipids. And a lot of scientists don't necessarily see them as organisms into themselves because there's certain parts of them that actually don't, they can't replicate on their own, right? They can't do certain things without host cells, right? So they might not be considered important enough to themselves, even though they're really interesting. So you have two different types of viruses for the most part. You have RNA viruses where you get rather unstable replication occurring, and you get DNA viruses. And mostly they're grouped into these families with the suffix viridae. So adenoviridae, herpesviridae, retroviridae. Some of those you might actually recognize. One thing that can occur in viruses, when we use influenza A as an example, is antigenic drift. So mutations can occur in genes that encode surface proteins, and these surface proteins can change the way that the virus looks to antibodies. So that's why basically the flu vaccine from a previous year is not going to be good to fight the flu this year. And in fact, scientists actually have to predict what they think the virus is going to look like to actually be able to create the flu vaccine and hope that it's going to be good against the flu virus for that particular winter. So in influenza A, you get these mutations occurring in genes that encode surface proteins, and there are 16 known HA subtypes and 9 known NA subtypes in influenza A. So all these different combinations can be very unique viruses. They can also make a change in surface proteins so that they become essentially invisible, or the host recognizes the viruses as themselves rather than an invader. So this is why antibodies from the previous flu can't fight infections, because the virus accumulates these mutations and they drift away from the ancestor strain, and you get this viral evolution of the common cold or the common flu. Another thing that can occur is something that's called antigenic shift, right? And I'm going to give another example using influenza A of how it's gone through this antigenic shift, which is a process by which two or more strains of viruses, in this case influenza, join to reassort into a new virus. Viruses often have about 8 segments of RNA in this case. Influenza but have space for 11 segments of RNA. So, evolution in action, they can take another 3 segments from a new virus. So H1N1, some of you guys are familiar with this, right? There was a small outbreak in 2009 when this virus was first discovered in 1970. At that time, there was 50 or 100 million people died from the H1N1 outbreak. And this happens because it causes a cytokine storm. So when the immune system is fighting pathogens, cytokines signal immune cells such as T cells and macrophages to travel to the site of infection. And the cytokines activate those cells, stimulating them to produce more cytokines. So this is a feedback loop that's normally kept in check by the body. But sometimes the reaction can become uncontrolled and too many immune cells are activated in a single place. So if this can be caused by exaggerated response by the body when the immune system encounters a new and highly pathogenic invader. In 1976, H1N1 started coming back and reported in Fort Dix in New Jersey, and 1 person died and 13 people got sick. And in 1977, 1978, this was called the Russian flu, and was an epidemic in that area at that time. And in 2009, primarily the epidemic was in Mexico, where you have the swine flu or H1N1, where it's actually a combination of 6 genes, which was a mixture of bird, swine, and human flu. So that's what made up this virus.
It turns out that this 2009 H1N1 began circulating in pigs in 1918. at the same time the 1918 influenza virus outbreak was occurring in humans. But because these two viruses were in different hosts, they evolved differently. So they both have a common ancestor going back to 1918, but because the swine H1N1 was in pigs, it evolved differently from human H1N1. And now that swine H1N1 is basically a new virus to humans, and the pre-existing immunity we have to seasonal H1N1 doesn't really protect us significantly from it. Whales and seals both carry particular strains of influenza. Horses carry influenza virus strains, and recently dogs have become a significant source of influenza virus infection. An influenza virus particle is roughly round in shape. It has a couple of protrusions coming off of it in two different shapes. One that's more of a spike and one more of a propeller shape. The H spike is used to enter a cell, and the other, the N spike that we always refer to, is used to actually get away from the cell. Anywhere from 9 to 12 hours later, the surface of that cell starts to form little bubbles, and those bubbles are new virus particles coming off. For about 3 to 4 hours, that cell will be able to make infectious virus particles, and it can make anywhere from 10,000 to nearly 1 million virus particles depending on the cell type. So if you're immune to infection, with influenza that means that you have antibodies that will bind to the virus particle and keep that virus particle from interacting with the cell.